Hello, my name is Christopher Ocampo, um, and today I'll be going over Dr. Gavara's presentation at the Brecker's virtual event discussing novel concepts in mechanobiology for Engineering 470. So just to go over what we're going to go over today, first I'll give a brief introduction to Dr. Gavara. Then I'll discuss a little bit about what AFM is. I'll talk about how Dr. Gavara uses AFM in her research and how we can utilize Dr. Gavara's techniques in the future. And then I will conclude this presentation. So who is Dr. Gavara? So she is currently a researcher at the University of Barcelona, specializing in biomedical engineering and biomaterials. She primarily studies the mechanical properties of living cells, and she's also an honorary senior lecturer at Queen Mary's University in London. So what is AFM? AFM, or atomic force microscopy, is a high resolution non-optical imaging technique. It uses a cantilevered sharp tip or round tip, depending on the type of tool you choose to utilize, to scan over a surface to get basically a rendering of the microsurface of the sample. As the tip approaches your sample, the magnetic force, the micromagnetic force, will influence deflection on the cantilever. There is a laser that is reflecting off of the cantilever that reflects into a photo sensor that collects the data and records the deflection in the cantilever. Here is a slide of that photo, the photo sensor. So the laser bounces off of the cantilever into this photo sensor and changes the voltage depending on the quadrant of where the laser hits, and that's how you can detect the deflection. Is it changes the voltage, and then you could calculate the deflection. So how does Dr. Gavara use AFM in her studies? So her methods are, so instead of scanning over, she does still scan, but she essentially probes her samples repetitively to record the reaction of the sample, depending on how much force it takes to basically indent the material or the sample. And depending on the instrument she uses, she can use that deflection to calculate things like a modulus of elasticity, among other properties. And these are the typical renderings you can get. So Dr. Gavara's focus is mainly on the cytoskeleton, which reveals both the structural and elastic properties of living organisms. Here are more scans. The data she collects can be utilized to show the difference in these properties throughout the sample. What she calls, we'll get to that later. She also goes over, let's go back. She also goes over the benefits and requirements when using AFM or calibrating your instruments to get the best data and most viable data. Some of the main capabilities is that you can create multi-layered mappings of your sample, which he calls multiplex imaging, so to speak. And another thing that she talks about is that you can get all this data very quickly, because you're essentially doing all the calculation on the back end, and you're essentially just collecting the data as you scan. Again, she drives home that with improved calibration and better use of the tooling, you can attain better data and record mechanical parameters to better capture your understanding of the biological nature of your samples. Oh, you could, and also she talks about how we can utilize machine learning with this data. So what can we do with this, with the methods that Dr. Gavara has been utilizing? So not only are we collecting renderings of our samples and of microorganisms essentially, but we can essentially gather even more data and better figure out essentially the nature of our samples. We can get a better understanding of behavior, of how certain parts of it migrate depending on how it's interacted with. We can utilize this data for better understanding of healing wounds or cancers, inflammation. Sports medicine can utilize this work as well, because you get a better understanding of how things like the ligaments react in certain situations. We can also use this data to help probe diseases, viruses, bacteria, to better understand how they react or how they behave, and also utilize this for discovering new drugs. Here's just another example of some cell partial recover or resolved cell mechanics. So it's another multiplexing experiment that she's done, where you can see that each of these graphs can illustrate a lot of things. Like you can get both at like a topographic reading, you get the modulus elasticity, viscosity, adhesion. You could record all of that within a single scan. So our conclusion, Dr. Gavara's methods of utilizing AFM for both imaging and data analysis of the mechanical properties of the cytoskeleton can pave the way for deeper understanding of microbiological structures. Soon we may be able to utilize her methods to develop new kinds of medicine, as well as analyze diseases in ways we have never before. And that concludes my presentation. Thank you.